My dear students, most welcome. Today we are going to start the very first chapter of class 12 and very important one, the introduction about the reproduction in organisms, which forms a basis for the next chapters. So the chapter is reproduction in organisms. When an organism second birth, the ultimate process, the death. So, from birth to the natural death of an organism, this duration is called lifespan. So, what is lifespan? The period from birth to the natural death of an organism is called its lifespan. Now, what is reproduction? Reproduction, re means again production to produce. So, it is defined as a biological process in which an organism gives rise to a young bird, that is offspring, similar to itself. They may be similar in genotype or phenotype or both. Further, reproduction is broadly classified into asexual reproduction and sexual reproduction. Okay? So, reproduction is broadly classified into asexual reproduction and sexual reproduction on the basis of involvement of gametes, formation of gametes, and fusion of gametes. Now, discuss one by one. What is asexual reproduction? Sexual reproduction essentially involves the formation and fusion of gametes. So this is the basic difference between asexual and sexual reproduction. Some other differences are asexual reproduction need only one parent, whereas sexual reproduction in most of the cases, 99.9% cases, requires two different type of gametes, that is male and female, or you can see two different parents, that is male and female parents. Now, we move with the asexual reproduction, and then in the next video we are going to discuss the sexual reproduction. Okay, so asexual reproduction is further classified in so these are some methods by which asexual reproduction takes place. Asexual reproduction takes place either by fission. Fission can further classify into binary fission and multiple fission. So this is asexual fission. Next is the fermentation. Asexual reproduction can also be experienced by the formation of birth, that is fermentation. By the reproduction. Next, by the behavior formation. This is how the are formed. We are going to discuss in detail one by one. Okay? Then by spore formation and vegetative propagation. Vegetative propagation further divided into natural vegetative propagation and artificial vegetative propagation. Now, let's start with the fission. Now, as we are going to discuss about the fission. Fission is either binary or multiple. Binary fission is either irregular type, transverse type, or longitudinal type. Irregular fission is most abundant type of fission in amoeba type of animals and organisms. For example, let's take the body of amoeba have nuclei, so many nuclei are there, and when there is plenty of food, it gets formation of nuclei and finally transverse binary fission is actually found in some specified protozoans and metazoans also. So one of the best example is paramecium and another is planaria. Actually planaria have highest capacity of regeneration. Now there are two nuclei, one get disappeared during the sexual reproduction. For further details, I'll make another video on this for the postgraduate and undergraduate students. Now what happens in the transverse one, that a plate begins to form here at the middle, and at the same time nuclei divided in equal amounts and move to the different sides, to the different poles. And when the plate becomes complete, the shape of paramecium assumes like this. Now it looks like this. Further, the nuclei get separated and finally two daughter paramecia are formed with a complete structures. Now these are two daughter paramecia, and hence two daughter paramecia are formed by the process of transverse binary fission. Now in longitudinal binary fission like Euglena, what happens that a plate transverse a longitude plate instead of transverse plate is formed, due to which two daughters are formed as like this. So we have to draw a structure of Euglena. This is Euglena. Now what happens, that the apical notch or like this groove is going to be deepened, and in the very first step this flagella removed, or you can say it's shed off its flagella, and then start deepening this portion and eventually reaches up to this point. So the two daughter cells are formed, and as it gets separated, the flagella reappears and its pure shape also appears like these structures. Okay, having flagella. So this is longitudinal binary fission by which Euglena is formed and it reproduces asexually, called longitudinal binary fission under asexual reproduction. Now what is multiple fission? Multiple fission is actually a device. Many of the organisms, protozoans like Plasmodium, Vivax, sometimes amoeba also, when there is unfavorable condition in the atmosphere, they reproduce by this method by producing very small nuclei parts and divide it into in multiples, like one nuclei divided into 2, 2 into 4, 4 into 8, 16 and up to 32, depends on the different strains and species of the Plasmodium. Sometimes it is limited up to 16 and sometimes it moves up to 32. Now why and how they do so? Consider this is a Plasmodium parasite. Now during unfavorable condition, they divide their nucleus and at the same time they oozes out the cytoplasmic content on the periphery. When cytoplasmic content reaches up to the periphery, when it becomes in the contact of air, it becomes tough, and this type of structure is known as cyst. So cyst formation actually takes place, and it becomes thicker and thicker. Sometimes this layer also contain structure known as chitin, as in the case of amoeba and some other species. And inside this structure, with reserved food material, so many nuclei are waiting for the onset of favorable condition. And whenever the favorable conditions going to be set, onset of favorable condition, then each of the nuclei get converted into the mature plasmodium, that is number 16 to 32. So this is the multiple fission, and this is the best method to tide the unfavorable conditions by the organisms like plasmodium and amoeba. Now the next type of asexual reproduction is, the next method is budding. Budding is the formation of bud-like structure in either single cell organism or multi-cell organism, and by the help of those structures, these animals or organisms, you can say, they are get divided. Since asexual reproduction involves a single parent, all the daughter forms are identical to their parents, and that's why sometimes we say that the daughters are clone to their parents. Clone to their parents. So clone word is used because they are morphologically and genetically similar to their parents. Now, budding in yeast, that is Saccharomyces. It is an fungi, you know, this is a fungi, Saccharomyces. Simplest type of bud formation forms in this structure, that is yeast, when cytoplasmic content oozes out and a bulge structure is formed here, and the nuclei part get embedded in this part by this way, and a newly formed bud is appeared at the top of this structure. When it becomes mature, means the bud becomes mature, it gets detached from the parent body and forms a daughter yeast. So this is the simplest form of budding. Now, in some animals like Hydra, this is the example, this is Hydra. In some animals, budding also takes place. But here budding is much more complex than the yeast one. Why it is so? Because yeast is a single cell organism, whereas the Hydra is multicellular organism. It is a complex multicellular organism. Okay. Now what happens? So this mouth-like structure is called hypostome. This is hypostome, and the finger-like projection are called tentacles. Tentacles. These are called tentacles. Now this is the base. This is the body proper, having different type of cells, consisting of different type of cells. And at the junction of this portion at the posterior side, a bud is going to be initiated or formed, and this eventually become mature with appear of hypostome and tentacles. Finally, a mature structure is formed. A mature structure is formed here. Now this is a mature bud. This is a mature bud. On maturation, it gets detached from the parent body and adjusts itself on the holdfast or some structure and become to grow. Now this is a mature, or you can say the daughter Hydra. So by this way, asexual reproduction in some specialized type of manner in yeast and Hydra takes place. The next one is regeneration, and as you know, regeneration is one of the most common method found in the planaria, which show the highest capability. And regeneration actually means, if you look the structure of planaria, so this is a planarian structure. This is not budding, please don't be confused with the structures here. Now we are going to talking about the structure which is known as planaria, and it is a regeneration. It shows highest capacity of regeneration. What happens when you cut these structures from anywhere? These all pieces get converted into a newer structure of planaria and grow into the baby planaria further, grow into the baby planaria. Okay, so this is also an example of asexual reproduction, and it is called regeneration. Okay, so this is called regeneration. Now next one is fragmentation, which is another common method. Fragmentation, simply when a plant body get fragmented, fragmentation. It is the very common method in lower plants like in algae, Eulothrix and Spirogyra, where a plant body when get detached from its parent's body, if it is broken down by this way, each part is converted into a complete structure and separately growing as newly formed individual. Separately growing as newly formed structures. Now, this is called fragmentation. Next one is by the gemule formation. Gemule is actually formed in some sponges, some calcareous sponges. You know about the sponges in class 11. Not spore formation, but the gemule formation. So during unfavorable conditions, some sponges divide their nuclei so many times, like in multiple fission, actually, like in multiple fission, but having a different structure, this type of round structure, where nuclei along with the reserve food material inhabits up to the long time, or you can say during the unfavorable conditions, it is closed by a thick mucous structure, and there are so many spines which are present in this to save this, to make a protective layer, and these are called spicules, calcareous structures. So this type of structure is called gemule, and at the onset of a favorable condition, each nuclei get converted into a full term sponge. Each nuclei is get converted into a full term sponge. So this is called gemule formation. Now next one and important one is by the spore formation. Spores are the special structure which is formed in most of the lower plants like algae and fungi. Spores, either they are motile or non-motile type. If they are motile, means if they have movement, they are termed as zoospores, means they have a movement. When they are non-motile, they are aplanospore, hypnospore, and so many other names are there for such type of spores. Now, motile type of spore, that is zoospore, actually have a flagella, or you can say sometimes they have two flagella, either single flagellum or two flagellum. And the best example for this type of structure is Chlamydomonas. In algae Chlamydomonas, this type of structure is found, where two different type of flagella are also found, Whipless and tensile type, you know it. And for non-motile spores, we have some common examples from fungus structure, that is fungi. And on the basis of their structure, of their formation, of their method, they are broadly classified into either they are sporangiospore or they are conidiospores. So when they are formed in an enclosed structure like this, in a particular enclosed structure, and on the maturation they are getting out from this structure, which is called sporangium, and that's why these spores are called sporangiospores. Okay, so now each of these spores get converted into new fungal plant body and growing up to its maximum limits. Another method by which these are formed, as in the case of penicillium and other some structures, they are arranging in this fashion, in this manner, like in this fashion, and they are growing towards up like this, forming a new structures of spores at the upper side and the mature one at the lower side, and this is known as what we call is the structure, and this is the best example of, this is the sporangiospore and this is conidiospores. Conidia like structures are formed here, and this is the conidium structure. This is conidia. Okay, so the best example of the conidia are aspergillus and penicillium. You can see these structures on the fungal structures. Okay, sorry for a little disturbance. So these are spores by which the lower simple animals and plants get reproduced asexually. Now, vegetative propagation. So, in very next lecture, we are going to discuss about the vegetative propagation, because it takes a lot of time. So, be ready. It may be tomorrow that we upload the lecture on the vegetative propagation. Now, a request. The whole world is suffering from a virus, or you can say a very severe condition, that is COVID-19. Please cooperate with the government and stay safe in the home. Continue your studies through online platforms, and I request you to please be safe and keep safe your families. Thank you.